Hi everyone, this is Shakil Ahmed, your biology educator, and I am here with one more topic of our Cambridge biology syllabus. 21, the biology paper with you. So let's start from here. Question number one, there are two types of cell, prokaryotic and eukaryotic. Bacterial cells are prokaryotic and plant cells are eukaryotic. There are differences in the structural features that are common to bacterial cells and the plant cells. For example, the cell surface membrane in a plant cell contains cholesterol, but in a bacterial cell, the membrane contains molecules known as hopenoids. Cholesterol and hopenoids have the same function. Some of the main structural features common to both types of the cell are shown in the table 1.1. Complete table 1.1 by giving one difference between a bacterial cell and a plant cell for each structural feature listed. The difference between the cell surface membranes of the two types of the cell has been completed for you. So one of the differences given over here. So this is the feature. So the first feature is the cell surface membrane. It has been already mentioned that the bacterial cell surface membrane contains hopenoids, whereas the plant cell surface membrane contains cholesterol. Now, coming to the next feature, which is the ribosomes. We know the ribosomes in a bacterial cell are the smaller ones, and we call them as the 70s ribosomes. Whereas the ribosomes in the cytoplasm of a plant cell are the 80s ribosome. Although a plant cell may also contain the 70s ribosomes, but inside the mitochondria and the chloroplast. DNA in a bacterial cell is free in cytoplasm. And we can also say that DNA is without the protein histone. Whereas DNA in the plant cell is associated with the protein histone and is enclosed in the nucleus. So we can say it is enclosed in nucleus of the cell. Now the cell wall of the bacterial cell is made up of the peptidoglycans. So cell wall is made up of peptidoglycans, whereas the cell wall of a plant cell is made up of the cellulose molecules, polysaccharide cellulose. So we have the 6 points for it. So each point should fetch us zero point 0.5 marks. That's why we have the 3 marks total for this part of the question. Now moving to the question number 1, B part. One role of the cell surface membrane of bacterial cells and plant cells is the transport of substances into and out of cells. Explain how membrane carrier proteins and membrane channel proteins are involved in the transport of substances into and out of the cells. So first of all, we know these proteins, carrier proteins and the channel proteins, they are actually used for the movement of the hydrophilic substances. The substances which are charged or hydrophilic um, will not be able to pass through the membrane because the membrane, overall the membrane is the hydrophobic. It contains the fatty acid chains which are hydrophobic. So for the movement of the charged molecules, we have these channels. So they allow the movement of the charged molecules or the ions. And number two is that channel proteins are gated proteins, means when they have to allow the movement of the ions through them, they will slightly open, allowing the movement of ions from the higher concentration to the lower concentration, the process that we call the facilitated diffusion. Whereas the carrier proteins are the proteins which can flip their shape. So these carrier proteins may use ATP molecules to flip their shape, and by using that ATP molecules, they can allow the active transport of the ions to take place. So this is a question for 3 marks. So let's write down the 3 points for this. So both the proteins allow the transport or movement of ions or charged molecules, or in other words we can say the hydrophilic molecules. So both have hydrophilic interior, which may be lined by the hydrophilic vesicles or the amino acids. So channel proteins are gated and allow facilitate diffusion to take place, whereas the carrier proteins can flip their shape. These carrier proteins may use ATP if active transport takes place through them, if active transport is taking place, takes place. So moving to the next part of this question, the part C of question number 1. Figure 1.1 is a photomicrograph showing chloroplasts in plant leaf cells. So we can clearly see the chloroplasts marked over here and the cells. Explain why the chloroplasts are seen only around the periphery, the edge of the plant cells. We know the main portion of the cytoplasm is occupied in the plant cells by the large vacuole. And when a plant cell is in the turgid condition, the vacuole is also swollen up. So it pushes the cytoplasm and the contents in it against the cell wall. So look, we can see the chloroplasts are also pushed against the cell wall. So this is why the large vacuole in swollen form pushes the chloroplasts to cell wall. Because the question for 1 mark only, so 1 point is sufficient to fetch us this 1 mark. Figure 1.1 shows plant cells in a root tip where cell division by mitosis is taking place. Identify two cells in the figure 1.1 that are in different stages of mitosis. Draw a label line to each cell and add the name of the stage of the mitosis that is shown by the cell. We can call this, we have to annotate this figure where we read the small details. So I can clearly see this cell stage is the metaphase. The metaphase is quite clear in it because the chromosomes are aligned at the equator. Chromosomes aligned at the equator of the cell or the center of the cell. Whereas this stage of the mitosis, we can say this is the prophase because we can clearly see that the nuclear membrane has disappeared, the nucleolus has disappeared, the chromosomes can be seen because when they condense in the prophase. Moving to question number 2. The high blood pressure at the arterial end of a capillary network results in some components of blood leaving the capillaries and forming tissue fluid. At the venous end, the presence of plasma proteins allows the movement of water by osmosis back into the capillaries. Figure 1.1 is a diagram showing a capillary network. The lymph vessels and the blood vessels at the arterial and the venous ends of the network are also shown. So we can clearly see it here. These are the body cells. These are the capillaries, and the tissue fluid is going from here. So this is how we can see it over there. Red blood cells and the plasma proteins such as albumin remain in the capillaries and are not found in the tissue fluid. So albumin and the other protein they remain in the capillaries because and they are not found in the tissue fluid. So they remain in the blood. Explain why red blood cells and the albumin do not leave the capillaries. So the question is why do they do not leave the capillaries? The answer to this one is because the proteins and this albumin as well as a protein, these are larger in size. Mostly the substances seep out of the capillary through the gaps in the endothelium of the capillaries. So endothelial lining is there which has the gaps in it. The molecules seep out of it or they leak out of the plasma through these. But the proteins, plasma proteins are large enough, they cannot pass through these endothelial gaps. So this is the reason why. So we can say that endothelium or endothelial gaps in capillaries are small and do not allow plasma proteins to leave capillaries. Now, question number 2 and part B. Name the chemical reagent or reagents used to test for proteins in a sample of blood plasma and state the color change that will be seen if protein is present. We know the test for the protein, we use a biuret reagent. And what's the result? If the protein is present, the positive test of the protein, it shows us the color change from blue, because biuret is blue in color, to purple or lilac. So if the color changes to purple or lilac, protein is present. If the color does not change, the protein is absent. Moving to question number 2, C part. When a person moves from sea level and stays at high altitude for a few months, there is an increase in the number of red blood cells per cubic millimeter of the blood. Explain why this increase occurs. So, as the person moves to the higher altitudes, the concentration of the oxygen decreases. So, oxygen being at the lower partial pressure, that means the saturation of the hemoglobin is not 100%. The saturation of the hemoglobin with the oxygen will decrease. So, the body carries less oxygen, so it can cause the problems in the person. So in order to make up for this lesser oxygen in the body, there is a natural tendency that the number of the red blood cells in the person living at the higher altitudes increases. We know there are other changes also that take place. They have the larger lungs, especially the right lung, which sends blood to the to the which actually receive the heart has the larger right ventricle, which sends blood to the lungs for aeration or oxygenation. And then also they have the broader chests to accommodate more larger lungs. So we have to here write down about the number of the red blood cells. So we see that at higher altitudes, the partial pressure of oxygen in the atmosphere decreases. So there is less saturation of hemoglobin with oxygen. The persons living at higher altitudes have increased number of red blood cells per cubic millimeter, so that there is increased number of red blood cells to carry more and more oxygen. So it will make up for the oxygen deficiency in the person. So red blood cells, they contain the hemoglobin, and hemoglobin, we have more number of the red blood cells, so more hemoglobin, so more saturation of the hemoglobin with oxygen. A low blood albumin concentration can lead to a condition which is known as the edema. Edema is a swelling of tissues caused by the accumulation of tissue fluid surrounding the body cells in the capillary network. Suggest explain, Suggest and explain how a low blood albumin concentration can lead to edema. So when we have a low blood concentration, albumin concentration, the concentration is related to the water potential. Means lesser the concentration, higher is the water potential. So when the fluid is lost, it usually returns to the blood at the capillary end. But when there is the higher water potential in the blood, that means the concentration gradient or the water potential gradient between the tissue fluid and the blood plasma is decreased. So less fluid comes back into the blood, because of which the fluid gets accumulated in the surrounding tissues and results in the edema. So we have a question for 3 marks, so we will write down the same thing. So low blood albumin increases water potential of blood. So less fluid, lost fluid returns to the capillaries at the arterial end as water potential gradient is reduced because of the low number of the albumin proteins. So fluid stays in tissues resulting in edema. Okay, now we have the part E of question number 2. Albumin transports some cell signaling molecules from the cells where they are synthesized to their target cells. The cell signaling molecules bind to specific cell surface membrane proteins on the target cells. Name the type of the cell membrane proteins to which the cell signaling molecules bind. So we call them as the receptor molecules or the receptor proteins. Receptor molecules which receive the cell signaling molecules or to which the cell signaling molecules. So moving on to question number 3. Baculovirus is a virus that kills some of the insect pests of major crops. When the virus is released to the outside of the insect body, it is contained within stable protective structures known as polyhedrons. The main component of the polyhedron is a protein molecule, polyhedrin. Polyhedrons can be sprayed onto plants as a bioinsecticide. They are ingested by feeding insect larvae, and once inside the insect gut, they break down to release the virus. So the first part and the E part of this question number 3 goes like this. Explain why the term infectious disease can be used to describe the effect of baculovirus on insects. And this question is for 2 marks. So first of all, we can say that how can we say it's the best infectious disease? So we can see it from here. But baculovirus is a pathogen. A pathogen is an organism that causes a disease. And if it is an infectious disease means it can be transmitted from one organism to another organism. So the disease caused by the baculovirus is an infectious disease which can be transmitted from an infected to a healthy individual or the organism. So we can see that baculovirus is a pathogen and can be transmitted from a healthy from an infected to an to a healthy organism. So we got the 2 points mentioned that it is the pathogen. It can be transmitted from healthy from an infected to a healthy organism. Now B part of this question number 3. Suggestion explain the conditions present in the insect gut that can cause the breakdown of polyhedrons. So we had to suggestion explain the conditions. As they mentioned at the beginning of this, at the start of this question, that these polyhedrons are the proteins. And we know that proteins are broken down by the proteases. And there are different types of the proteases that work in the different conditions. So proteases are the enzymes. Obviously, their activity will be affected by the pH of the medium in which they work. And the enzyme activity is also affected by the temperature. So this is actually the conditions. So state and suggest so polyhedrons will be broken down by proteases, and these proteases that can work in acidic or alkaline conditions. Like we have pepsin that works in the acidic conditions, whereas the trypsin works in the alkaline conditions. Both of them are the proteases. Secondly, we need that the suitable temperature is required for optimum functioning of the enzymes. So these protease enzymes, they will work in the temperature, which is usually most of the enzymes, they have the optimum temperature from between 35 to 40 degrees Celsius. Now, question number C part of this. A polyhedron molecule is composed of three identical polypeptides. Each polypeptide is 245 amino acids long. So these are three identical polypeptides. The first 10 amino acids of the polypeptide are shown in the figure 3.1. Now, figure 3.2 is a ribbon diagram of a single polypeptide. Figure 3.2b shows a polyhedron molecule composed of three polypeptides. The two diagrams are not to the same scale. So, what is the question? The question here goes with reference to figure 3.1 and figure 3.2, describe the structure of a polyhedron molecule. So, we can clearly see it here that the polypeptide or the molecule consists of three polypeptides. So it has a primary structure. Primary structure will determine the amino acids and their sequence in it. It has the secondary structure that we can clearly see it here. It has the alpha helices and the beta pileated sheets. The tertiary structure depends upon how the three polypeptides are folded to give it a three-dimensional shape. And it has a quaternary structure as well because it is made up of more than one polypeptide. So we have this question for 4 marks. So we got the 4 marks to get full 4 points to get the full mark. So first of all, it has a primary structure and that determines the amino acids and their sequence in it and their sequence in it. Then each polypeptide has a secondary structure that can be seen made of alpha helices and beta pileated sheets. Then the three polypeptides are folded to give it a tertiary structure. The protein has quaternary structure as it is made of more than one polypeptide. Part D of this question number 3. The sequence of DNA nucleotides for the gene in baculovirus that codes for the polyhedron polypeptide has been determined. Explain why the amino acid sequence of the polypeptide cannot be used to deduce this sequence of nucleotides in the gene that codes for the polyhedron molecule. So, if we know the sequence of amino acids in the protein, can this sequence of amino acids we use to determine the sequence of the of the nucleotides in the DNA molecule? We cannot do that. Why? The reason is each amino acid is coded for by more than one codon. We have different codons, more than one codons that are used for or coded for each amino acid. Like we have 64 codons that code for 20 different types of the amino acids. So we may not know which codon is present in the DNA. Secondly, the protein includes only the amino acids which are coded by the exons, whereas the introns have been removed from it by in the DNA splicing or DNA processing in the nucleus. So also we cannot, from the amino acid sequence, we cannot actually mention the sequence of the introns in the DNA molecule. So these are the reasons that we can also we can say that amino acids are coded for by codons, and each amino acid has more than one codon coding for it. So which particular codon is present in the DNA can not be ascertained. Secondly, the protein contains only amino acids coded for by exons, as introns are removed in DNA splicing. Okay, so moving on to question number 4. Barlinca is a variety of the common grapevine, Vitis vinifera. Barlinga grapes are used for making wine and are sold as fruit. There are economic and ecological benefits from using less water to irrigate grapevine plants while still producing a high crop yield. The rate of flow of sap within xylem vessels from roots to leaves can be used as an estimate of the rate of transpiration. This also indicates water uptake. Now, question number 4, part A of it asks us the hydrogen bonding of water molecules is important in the transport of sap within sap vessels, within xylem vessels. State the terms used to describe water molecules sticking together within the xylem vessels. We know the force of attraction between the same type of the molecules is known as the cohesive force. So, this is the term used to describe the cohesive force between the water molecules within the xylem vessels. Then, water molecules sticking to the cellulose molecules in the lining of the xylem vessels. So we know the force of attraction between the molecules of different types. Here it's the water and the xylem vessels. We call that as the adhesive force or adhesive attraction. Now, B part of it. An experiment was carried out to investigate the effect of leaf area on the rate of flow of the xylem sap during fruit development. The flow rate was measured over a 3-day period in 3 Barlinka grapevine plants with different total leaf area. The plants were growing in the same conditions. The results are shown in figure 4.1. We can see here that this dotted line is the leaf area grapevine 1, which has an area of 13.7 square meters. Then we have the completely dotted line leaf area grapevine 2, 9.6 square meters is the area. And the solid line is the leaf area grapevine 3 with an area of 3.2 square meters. So on the y-axis, we have the flow rate of xylem sap, which is in the centimeter cube per plant per hour. And this is the time of the day, or these are the number of the days on the y-axis. So this one is for the grapevine 1, which has the area of 13.7 square meters. This is for the grapevine 2, which has an area of the 9.2 square meters. And this one is for the grapevine 3, which has an area of 3.2 square meters. So the question goes like this way. The general pattern of results in figure 4.1 is the same for the 3 Barlinka grapevines. Explain why the general pattern of results is the same. Why is the general pattern of results same? We have a question for 2 marks. So obviously we can say that the 3 types of the Barlinka plants are growing in the same conditions. So any change in the factor in the conditions will affect the water loss equally. So that's why we have the same pattern shown by them. So we can say there's the same pattern as the grapevines are grown in same conditions. So any change in a factor of conditions will have same effect on all the grapevines. Now, part 2 of this question. With reference to figure 4.1, explain the differences in the results between the three grapevines 1, 2, and 3. Again, the question is for 3 marks. So we can see that most of the high flow rate is present in the grapevine 1, which has the high surface area. And then it comes the grapevine 2, which has a surface area lesser than the grapevine 1. And then the least flow rate of the xylem sap is in the grapevine 3, which has the least surface area. That means more the surface area, higher is the flow rate of xylem sap. Less the surface area, lesser is the flow rate of sap. So we can see this is the differences. We have to explain the differences as well. So higher the surface area of grapevines, then the expression means higher will be the number of stomata. So higher is the flow rate in xylem. So we can quote some data from the graph. So we can say in grapevine 1, it is the maximum flow rate is around, where has it gone? Come on, wait. Yeah. So in the one we have the maximum flow rate is here. It is between this is 350. So 10, 20, 30, 40, 50. So this is 330 per cubic centimeter, whereas the for the 2 it is 210, and for grapevine 3 it is 160 per square centimeter. So we can quote this data to get the full marks for this question. Then part 3 of this question, suggest an explanation for the decrease in flow rate of xylem sap shown at the time p in the figure 4.1. So going back to the figure, we can see that at the p, there is a decrease in the flow rate of the xylem sap. Now we will see what are the factors that decrease the rate of transpiration. So the main factor that decrease the rate of transpiration is the humidity. If the humidity increases in the atmosphere, that will decrease the water potential gradient or the water concentration gradient of the water molecules, and so lesser rate of transpiration flow rate in the xylem will be there. So suggestion explanation for the decrease. So higher humidity or atmospheric humidity in atmosphere will decrease concentration gradient, hence less flow rate. Outline how would you determine the surface area of one side of a leaf. So if we have a leaf, how can we determine the surface area on the one side? So we put a squared paper on it. This squared paper has small squares, so we can count the number of squares that cover the leaf surface. We'll count those which are filled more than half by the leaf, and we will exclude those which are covering less than which are being covered by less than half by the leaf. Then the total surface area we can divide by 2. That will give us the leaf surface area of one side. So determine how we do determine the surface area of one side of a leaf. So cover the leaf with squared paper and count the number of small squares covering leaf surface. So we can say count squares covered half or more than half of the by the leaf and divide by 2. Okay.